Bismillah Rahman Rahim. Assalamu alaikum to all. I hope my voice is clear and the screen is visible. I am Dr. Hamza Khan, resident hematology at the Indus Hospital. I welcome you all to the scientific session titled Blood Safety Connecting the Dots. In this session, we have a distinguished panel of speakers who will be discussing different aspects regarding transfusion medicine. So, without further delay, I would like to welcome my first speaker, Dr. Uzma Atta, a clinical pathologist and manager, Indus Hospital Blood Center. She has been associated with Indus Hospital Blood Center right from its inception since 2013 and is exclusively involved in training and development of blood donor collection and mobilization teams. She will be discussing regarding Indus's experience in voluntary blood donation during the COVID 19 pandemic. So, Dr. Uzma, please, you may start.
Assalamu alaikum. My name is Dr. Uzma. I'm the clinical pathologist and the manager of Indus Hospital Blood Center. The presentation topic today is the voluntary blood donation and COVID-19 experience, which was faced by the IHH Blood Center. Worldwide, the blood supply faced unprecedented situation with a multitude of blood drives cancelled as the organizations, educational institutes were temporarily closed due to pandemic, and countless patients across the world continue to rely, those who were continue to rely on the blood donors to provide life-saving components were all compromised. So similarly, the situation in Pakistan were worsened due to lockdown, as the donors were reluctant to expose themselves from outside worlds and getting the deadly disease. With the advent of COVID-19 in Pakistan, the already compromised and fragmented transfusion service received a severe blow, putting the lives of transfusion-dependent patients, especially those of thalassemia and cancers. Actually, it impacted the supply of blood to the thalassemia patients, especially the fear of the virus contraction coupled with lockdown and restricted mobility disrupted the entire transfusion chain. To cater all these problems, which were the blood centers across the world were facing, WHO actually developed the guidelines for blood donations during the pandemic, in which the guidelines recommends that the mitigating the potential risk of transmission through blood transfusions, staff risks, donor exposure to COVID-19, as well as the risk of reduced availability of blood donors. They also addressed managing how to manage the demand, ensuring undisrupted supply of critical materials and equipment, and communicating to ensure the donors, recipient, all staff, relevant stakeholder populations should be properly informed. ABB and FDA, the American Association of Blood Banks, and the FDA also developed a task force to address the issues related to blood shortages. They issued a guidelines statement to address the shortage. This emphasizes the patients is not at the risk of connecting the disease through the blood transfusions. In addition, blood centers were strictly requested to follow SOPs related to COVID-19 and to ensure the safety of the patients and encourage them to donate blood. The biggest challenge for the blood centers across the world was to retain consistent supply of blood to the hospitals. This was same faced by Indus Hospital Blood Center. The blood center inventory was exhausted as the blood drive were cancelled due to lockdown. Since the blood center is mostly relying on its voluntary blood donation, it was highly impacted as the large number of blood drives were cancelled. Especially the major was educational institute and the corporate sector, which were all closed, and the lockdown was imposed. Donors was restricted to move as the lockdown was there, and the fear of the donors on getting exposed to the phlebotomist and to the outside world was also there. So these are the contributing factors which actually severely impacted that the donors were the turn of the donors were quite low. If you could see the trend and effects of the blood donation due to COVID-19, there was a remarkable decrease in the blood collection noticed in the during the pandemic, and there was a poor response from the donors and the low turnover of the blood drives and the donor center despite the multiple requests. till date, few sites, we are still struggling with the few sites to get the donors on, because they themselves are struggling with their financial issues, and they're laying over of their own employees, which is the setback due to the lockdown and the pandemic, which is like affecting entire world, hence resulting the lack of potential donations. So you could see that from 2000, we have presented this data from 2013, since the inception of Indus Blood Center. There is an increasing trend till 2019, and at the end of the 2019, the blood donations started decreasing, and still we are facing a decreasing trend, the blood donations. It has never been regained yet. This data actually represent the blood drives versus the collections on the drives. And if you could see that the blood drives are, the average blood drive basically was 70 to 75% per month before the pandemic, and the average collection per day was like 70 to 90 units per day. And they were including the large camps and the small camps. There was a mix of camp. With pandemic, the number of blood drives were greatly impacted, with mostly collection was from small camps to get the optimal blood supply. uh, Actually, there was a restriction that the large crowd cannot be gathered. So the blood drives were mostly consisted of small blood camps. A good collection can be observed in the month of April. If you could see that there is a increasing trend of 2006 donors, despite lockdown, and only 46 blood drives. This was actually uh, during the strict lockdown, which was imposed in Karachi. This was largely due to the community contribution and strong support by a religious group who called their countrywide followers to came forward and make their blood donations, as much as blood donation as possible. So that this actually was the impact of the community support and the religious group. We can see that. Later on, if you could see that in September, there was a rise in the camps, but still, like, there is like 100,000, between 1,000 to 1,200. There is a like a stagnant kind of a trend in blood donations. Similarly, in 2013, quite a unpredictable kind of a trend, low blood drive ratio with better collection, but not as optimal as it should be. So still a mixed trend and significant number of blood drives, but suboptimal collections comparatively can be seen. This is actually a slide in which I just wanted to explain um, the contribution of different sectors of the society towards voluntary blood donations. In 2019, the highest contribution was made by educational sector and the factories, the production factories, the mills, and everywhere. So in 2013, since the lockdown was imposed, only 41, that was actually before the lockdown was imposed. So the educational institutions were only 41. The factories goes down to 62, and the Islamic organizations were like 25, which is like a good trend number of donations were made from them. And the rest were almost similar, except the residential area is on the higher trend, because we actually called out the communities and individual donors to donate. So the residential areas and the neighborhood donors were quite on the rise. So you can see that in the residential area, in 214 donations were made at the residential area. How did we actually cater this situation? So different strategies were planned to cater the shortages. To overcome the hurdles, the blood center devised a strategy to contact potential blood donors through social media, WhatsApp group, like individual donor calling, convincing them to donate blood, telling them about the scarcity of blood, calling out group of pledged donors via email, social media, Facebook, uh, Instagram, Twitter, whatever was available to us. A comprehensive strategy was also implemented to collect blood from the doorsteps of the donors. So majority of them at that time was home collections. Community and volunteer group mobilizations, they were contacted. There were students of alumni. There were like few people from certain community, even from the neighborhood, and even from some of the people called as voluntarily, that they have group of people who can donate during this lockdown. So they actually contacted and they actually contributed a lot in this period. For the execution, a proper execution was done. We drafted an SOP keeping in view all the pandemic situation that was approved by the Infectious Disease Department of Indus Hospital and the Medical Directorate. Then later, we just sent this to acquire permission letter from the provincial government to move our collecting units during the lockdown for blood donations. The precautionary measures taken by the staff was the team was screened during the COVID before leaving. They all get used to screen verbally. They had to be like screened as if they are having a fever or if they're having any symptoms or if they're having temperature. So before leaving, all entire staff was screened for COVID-19. The camp setup limited to the group of 8 donors at the time to avoid grouping. A distance of at least 4 to 6 feet for maintained. Staff don't proper gears and follow all the SOPs, especially drafted for the pandemic. The mobile units in which we were collecting blood was properly disinfected before and after each drive, and the donor couches were also disinfected after every draw. The camp layouts and logistics were planned as per the drafted SOP, and a significant distance were maintained. There was a risk assessment tool given to us by the Infectious Disease Department, which consists of few questions regarding the sign and symptoms and the traveling history of the donor, so that we can just screen them out if the donor is exposed or having or could have COVID-19. Maintaining significant distance at the campsite between the couches and the waiting area was also maintained. Donors were usually called by appointment just to avoid the crowding. And we tried that the setup should be done in an open area so that the closed area contamination could not take place. Alternate beds of the mobile bus or mobile van units were used. For example, we have like 4 couches in a mobile bus unit, but we used only 2 beds diagonally to draw the blood. And the donors were not like closely laying for the drawing of blood. They were like at a distance, and only one phlebotomist was there to attend them at a time, so that the crowding was not there. A significant distance between the couch, it was like an in-house camp. So we tried to maintain a quite at least a 5 to 6 feet distance between 2 couches. Our blood donors bestowed their trust and gratitude towards the through social media, that they had a great experience and they were fully satisfied with the precaution measures in this hospital took during the blood collection process, and we were really pleased that they show their satisfaction and their trust on us. So this is how we actually collected our blood during pandemic, and we are still following the same SOPs and same precautions. We are still struggling with the blood donation just because the pandemic situations is yet not over. And the corporate sector, though there are like contributions of corporate sector educational is started, but still they are struggling with the number of donors, with their presence of employees, because there are quite a layovers during the pandemic. So, and their financial situation also impacted a lot. So just because of these all factors, we are still struggling with our regular blood donations, but it's getting better. And we hope that in next year, it will get better more. So thank you very much.
Thank you very much, Dr. Uzma, for enlightening us regarding the experience. The next speaker is Dr. Neelum Mansoor, consultant hematologist at the Indus Hospital, proficient member of hematology faculty at Indus, and working as in charge blood center. She will be talking about good manufacturing practices in blood banking. So, Dr. Neelam, over to you.
Assalamu alaikum. I am Dr. Neelum Mansoor, and today I am going to talk about good manufacturing practices in blood banking. And it is not a very famous topic in our settings, so it will be a little boring for you. I have nothing to disclose. In this slide, there is a brief outline of my today's presentation, and I'll be talking about a basic concept of GMP and a brief history, its requirements, implications in blood transfusion services, GMP principles, and some recommendations by regulatory authorities, a brief summary of gap analysis of Indus Hospital and Health Network blood transfusion services, and some achievements of Indus Hospital blood transfusion services, and current status of GMP and transfusion practices in Pakistan. So what is GMP? As I told you, this is not a very famous terminology here in our settings. So I just put in here a small or simple definition of GMP. It is actually a set of regulations, codes, and guidelines for the manufacturing of drugs, substances, and drug products, medical devices, diagnostic products, and foods, and biological products. In biological products, actually, it comprised of blood and its components. So these are the regulations which describe minimum rules for the companies to assure quality, security, purity, and effectiveness of the products. GMPs are basically found in Title 21 Code of CFR. I'm talking about FDA regulations. And if we particularly talk about blood establishments, it is mentioned under CFR 606. These are the 5 famous P's of good manufacturing practices, which includes a well-defined roles and responsibilities of people, specifications at every phase of production of products, well-documented processes, and procedures defined for critical processes, cleanliness of equipment and calibration of equipment, and cleanliness of premises. So if you talk about the history of GMP, it is itself a very huge topic. I just put some important facts over here. Basically, GMP started in 1902 with the Biological Control Act, that was first federal law affecting domestic drugs. And then in 1906, Pure Food and Drug Act was announced, and FDA is created in 1927. In 1941, there was a tragic incident happened about sulfathiazole tablets that were contaminated with phenobarbital, and many people died in that incident. So at that time, the GMP was initiated, and another tragedy happened in between 1958 to 1962. As we know that thalidomide was available over the counter and no prescription was required for this particular drug, and thousands of babies born without limbs and other birth defects. So at that time, proof of efficacy law was implemented. And then in 1963, GMPs for drugs were established, and in 1975, GMPs for blood and components final rule was announced. And it has been modified many times by different regulatory authorities, and it is very well described now. So if you talk about GMP in blood banking, it is basically a requirement that is usually set out in codes of GMP, developed by blood services in conjunction with regulatory or government authorities. The adoption of GMP provides confidence that a blood services component will meet the specifications of suitable existing standards. Whatever the standards you are following, whether it is AABB, WHO, FDA, ISO, whatever you are following, your standards should meet the defined specifications of that particular standard. So if you talk about implications of GMP in blood banking, it actually it does not mean manufacturing. And it is not specifically applied to the manufacturing of components or manufacturing of cells or manufacturing of reagents. It is basically cover all aspects of blood banking, starting from the recruitment of the donors, their screening, component preparation, storage, each and every step of testing, quality control, product issue, look back, each and everything is covered under GMP. So these are the basic 10 principles of GMP, which includes quality management system, personnel, documentation, premises, and equipment, quality controls and service contracts and analysis, complaints, non-conformance, blood component recall, audits, and validation and qualification of equipments. So these are the key principles of GMP that was announced by European Commission in 2006. These are more or less same as I described in the previous slide. And they also describe a flow chart, how to implement, how to plan, how to perform the GMPs, how to keep a check over it, and how to improve those practices. So this is the first and the most important part of the GMP, that is quality management system. It is a very huge terminology, and I just try to simplify it here. It actually satisfy regulatory requirements. It owned and understood by the workforce. It is an integral part of how everyone works, and it ensures the delivery of high quality services, and it should be GMP focused. QMS covers all these parameters like quality incident reports, self-inspection, technical agreements, complaints, component recall, traceability, adverse reactions, reporting, and continuous improvement, a very well formatted and well organized quality manual, and the document control system that is a very important part of QMS. These are important documents, SOPs, and guidelines that should be in place under the terms of QMS. Here, I just tried to explain the relationship of all these terminologies. So the quality management system actually defined the overall policy of a system towards quality. And quality assurance is a concept that ensures the policy is achieved. GMP basically deals with a risk that cannot be tested. It brings quality into the product, and quality control is a part of GMP that is focused on testing of material and products. And the most important thing is that quality management system should be under the supervision of a designated person who reports to the executive management. He or she should be properly empowered to run this system. Personnels, there should be competent and appropriately qualified personnel should be available in sufficient numbers. Their responsibilities should be well defined and documented and understood by them, and the staff appropriately trained should be authorized to perform those procedures, and their training records should be available in the department. Regarding training and competence, initial and continuous training in accordance to specific tasks should be in place, and training should be carried out by trained personnel, and there should be pre-arranged written programs, careful evaluation of competency, and continuous monitoring, and the need of reassessment should be documented, and training records and competency records should be traceable. This is a flowchart how to implement training and competence in the departments, and how to undertake a training needs its assessment, and the source of teaching materials should be defined, the teaching method should be there, and the periodic competency timing should be defined and clear to the staff, and the evaluation of the training program should be there. Regarding documentation, it is a very important principle, and it applies to all SOPs, guidelines, trainings, and reporting forms, from quality manual to procedures to instructions. Any forms and formats all should be clear, concise, and unambiguous, and it should be regularly reviewed and controlled, and they must be traceable, and all staff are well aware of all these documents. So only official copies should be available in the department, and any alteration to any of the records should be signed, and should always be done by an authorized person. Regarding production and procedures, it is a very large component, and I just summarize it over here, that it must follow clearly defined procedures, which should only be performed by trained and competent staff. And any significant amendments to the production process, whether equipment or materials, which may be affect the quality of service, should be validated. Validation studies should be conducted to show that the process, equipment, and any activity that leads to the expected results should be in place. And there should be a formal documented system for change control. And all changes that affect the product quality or reproducibility of the process should be formally requested, documented, and accepted. Premises should be well designed and constructed to ensure the safety, to minimize the risk of errors, and the layout, design, and operations must be according to the functions of the blood transfusion services. And the important thing is the well-defined storage areas, production areas, and access control should be there. Regarding equipments, equipment should be adequately specified prior to purchase. All equipment should have an original identifier such as serial numbers, and it should be validated correctly before being put into use. And when in use, its IQ, OQ, and PQ should be revalidated at regular intervals. Equipment should be calibrated and maintained according to written instructions, and defective equipment should be ideally be removed from the area. And if it is not possible to remove that instrument, then there should be a proper labeling or tagging should be done that the particular instrument is out of order, is not functioning. Regarding service controls and analysis, all tasks that have an influence on the quality of service and are performed externally by another party should be subject to a specific written contract. Prior assessment for the suitability and competence of the other party to carry out the activity or provide the material using a defined supply chain. Responsibilities and communication process should be well defined, and all the contracts by both parties should be signed by the quality assurance representatives. Regarding complaints, all complaints concerning potentially defective products must be reviewed according to the written procedures. There should be a recall procedure in place, and the recall operations must be able to be initiated properly and at any time. And the system to ensure rapid recall of any product found to be defective. Sorry, इसको दोबारा करना. Regarding complaints, all complaints concerning potentially defective products must be reviewed according to written procedures. There should be written recall procedures that are updated as necessary, and procedure of recall should be available and be able to be initiated promptly at any time. And full traceability of all products should be in place. Self-inspection and audits are planned independent investigation of selected elements of a quality assurance system to collect objective evidence of system's effectiveness. Internal audits are conducted to monitor the implementation and compliance with GMP. All audit results should be documented and reported to the management. Appropriate corrective and preventive actions should be taken in a timely and effective manner. So internal audits are actually self-inspection, and it is not an alternative to the official inspections that are done by official authorities or government bodies or any other regulatory bodies. And also, unfortunately, we do not have that particular kind of system implemented right now. Regarding validation and qualification. So what is validation? Actually, it is action of proving in accordance with the principles of GMP that any procedure, process, equipment, material, activity, or system actually leads to the expected results. While qualification is action of proving that any premises, systems, and items of equipment work correctly and actually leads to the expected result. Essential parts of GMP prove that critical aspects of work are controlled. Significant changes to the facilities, equipment, and processes which may affect the quality of the product should be validated. So this is the process qualification, process validation guidance, and it is comprised of 3 stages. That is process design and process qualification, and that is followed by the continuous process verification. So this is a equipment validation plan, and if you follow and if you actually follow the steps, it actually increase the confidence and reliability of the equipment and minimize the risk. In this slide, I just share some brief definitions of IQ, OQ, and PQ. And regarding IQ, it should be performed on new or modified facilities, systems, and equipment. It confirms that the equipment has been installed correctly, and OQ establishes that the equipment is fit for the purpose. And regarding performance qualification, it demonstrates that the equipment, system, or process can consistently produce the required quality. And it is very important to perform and define a regular interval of all these parameters. Regarding electronic data processing, records of the following should be maintained like, sorry, so electronic data processing, records of the validation of system software, hardware, database, and electronic data transfer should be in place. Fulfillment of applicable lifecycle requirements for internally developed software should be available, and numerical designation of system versions, if applicable, with inclusive of dates should be in use, and monitoring of data integrity for critical data. Sorry, sorry. Electronic data processing, records of the validation system, software, hardware, database, and electronic data transfer should be in place. Fulfillment of applicable lifecycle requirements for internally developed software should be available, and numerical designation of the system versions should be in place. An alternate system should be maintained to ensure continuous operation if computer-assisted functions are unavailable. And a system designed to prevent unauthorized access to computers and electronic records should be established and followed. And it is very important, particularly in terms of the electronic data processing and the kind of softwares implemented nowadays in our departments. So GMP in a nutshell is actually, it is all about well-trained staff, documentation, good premises, and quality control is all about testing materials, while quality assurance is all about testing processes. So these are the GMP principles I described in my previous slides, and you can see that all international regulatory authorities recommend these principles, whether it is WHO, AABB, European Commission, FDA. All these regulatory authorities recommend these principles with some minor differences or some weightage to one or two particular clause, but almost they all recommend these parameters. So on the basis of all these principles, we performed a gap analysis of our own system. And here in these slides, I'm going to present a brief summary of that gap analysis. So you can see in quality management system, we are almost achieving around 70% of the required principles, and there are some deficiencies. Some few of the systems are partially available, while one or two are not available at all. Regarding personnel and documentation, we are meeting 100% requirements. Regarding premises, unfortunately, we are meeting 50% of the requirements recommended by the international authorities. And equipment, computerized systems, qualification and validation, and management of materials and reagents. In all these principles, alhamdulillah, we are meeting 100% of the requirements as the system is running under a great leadership and the team is very well trained and competent. So alhamdulillah, we are meeting the most of the requirements under all these principles. Manufacturing is the major part of the GMP, and in this particular principle, we are meeting around 80-85% of the requirements. In contract manufacturing analysis, almost 70% of the requirements are fulfilled. So here in this slide, I just summarize the gap analysis we performed, and I'm sharing the major deficiencies identified, which includes the quality management system. And in this particular principle, the product recall and look back policy is not available. So it should be defined and implemented in the system. Risk management, deviation evaluation, and change control. All these parameters are lacking, and it needs improvement in documentation. Regarding premises, design and construction is not according to standards, and lack of separate production area is a major challenge, as well as insufficient storage area, especially when we are talking about centralized wet banking and we have a huge number of donors and a huge blood collection. So we need to have well demarcated production and storage areas with a very good access control. So hopefully all these deficiencies will be addressed in new premises, which is already under construction. Regarding manufacturing, donor registry is a major lacking, and donor surveillance is also not available. So hopefully donor registry is already in process, and it will help in the surveillance program as well. Uh, Donor apheresis is also not started yet, and leukocyte reduction and product irradiation. All these principles are still lacking. But I would share that we have successfully started therapeutic apheresis, and hopefully we will be moving on to donor apheresis very soon, as the staff is already trained and the machines are already there, so we can start it at any time. Product irradiation, this issue has also addressed, as we have already purchased irradiator and the installation is already in process, while leukocyte reduction is still a challenge due to cost issues. These are some of the achievements of Indus Hospital blood transfusion services. The most important thing, that is centralized blood banking, and the dedicated donor mobilization team, our mobile drives, a huge pool of voluntary non-remunerated blood donors, a very well designed and robust blood bank management information system, and we have a very well developed electronic document management system, the licensing for ISBT 128 for the labeling of blood products, implementation of confirmatory and supplementary testing for TTI, that is according to the international standards, and the algorithms are very well designed as per the international guidelines. We have successfully implemented platelet culture on 100% donations. Donor antibody screening has also been started. We have a very well established hospital transfusion committee, which has representatives from all the specialties of the hospital. Irradiator, I already told you regarding it, and we are in process of ISO 15189. Final audit has been done, and hopefully we will get the feedback very soon. So the future directives are all these lackings, particularly the donor registry, community engagement for the retention of voluntary donors, electronic cross match. We are in process of implementing it, and the product blood bank reagent manufacturing and the plasma fractionation is also in our to-do list. So here in this slide, I just share current status of the transfusion services in Pakistan. As we all know, it is fragmented and demand-driven system. Services are mainly provided by the public or private hospital blood banks, thalassemia centers, state-owned blood banks, and unregistered small labs. So there is lack of proper pre-donation screening, very poor quality of TTI testing, especially blood group. Even the blood grouping is not properly and completely performed by most of these centers, and the lack of documentation. This is the legislative framework of blood transfusion services in Pakistan, and it is started in 1997 when the first and the 10th Transfusion of Safe Blood Act was introduced. And since then, many amendments has been made, and the last amendment was made in 2016 and 17 in Punjab and Sindh Blood Transfusion Acts. So these are the blood transfusion service reforms and GMP in Pakistan. Sorry. So some major reforms took place in 2008 when blood safety reforms were initiated through Pak-German Joint Project. And in 2010, Ministry of Health initiated Safe Blood Transfusion Program at the national and provincial levels. So National Blood Policy and Strategy Framework was done by SBTP in 2014, and they designed national guidelines for the quality control and clinical use of blood products. And infrastructure reforms like constructing and a network of regional blood centers has been established. There was a survey conducted by SBTP in collaboration with WHO. It was not mainly based on GMP, and the purpose of the survey was not GMP. They basically targeted the screening of the TTI testing, but it was covering to some extent of GMPs. And if we see the results of their survey, they are not very encouraging. Anyways, these reforms and efforts in the last decade has propelled the need of safe blood transfusion at national level. Yet a huge commitment is needed at the level of community and government both. and it should be implemented all the principles of GMP and the transfusion practices. Sorry, इसको भी दोबारा देते हैं. Yet a huge commitment is needed at the level of both community and government to implement all the principles of GMP in the transfusion services. So, to the take-home message is how we can implement GMP in Pakistan is by designing some effective legal and regulatory framework, and to achieve 100, it is very important to achieve 100% registration of blood banks, ensure the compliance with policies and SOPs which are somehow prepared at the national and provincial levels, but not implemented very well or partially implemented in some places. We need to expand our centralized blood banking system. We also need funding and capacity building in terms of education programs and training of technologists and physicians as well. A huge campaign is required for the community awareness in terms of safety of blood, for understanding of TTIs, and for voluntary blood donations as well. So by doing all these efforts, we can move towards accreditation with regulatory authorities. Thank you so much. And I would like to thank ICON team for giving me this opportunity. And I must say thanks to Dr. Saba. And I really feel honored to represent Indus Hospital Blood Transfusion Services over here on this forum as in charge. And I would also like to thank Dr. Naeem Jabbar for his contribution in this presentation. Thank you so much.
Thank you very much, Dr. Neelam. That was in detailed discussion regarding GMPs. I hope the participants are getting along with us. If you have any questions regarding this talk, you are highly welcome. You may ask the questions by using the chat box. Okay, so we will move forward. Our next speaker is Dr. Fatima Meraj, consultant hematologist and section head hematology at the Indus Hospital. Senior hematologist having particular interest in MRD, flow cytometry, and blood banking. So, Dr. Fatima, you may start.
And Blood Center at Indus Hospital and Health Network. My today's topic is transfusion transmitted infections, reappraisal of blood safety measures. As we all know, accurate testing of blood donation plays a pivotal role in the provision of safe blood for the transfusion. The objective of the blood screening is to detect the markers of infection, so to avoid the transfusion of transfusion transmitted infections to the patients. So my presentation will cover the testings that should be carried out on every unit of the donated blood. This is the outline of my presentation. I'll briefly discuss the basics of the transfusion transmitted infections, something about the window period, and in detail testing algorithm, which will encompass both the screening and confirmatory testing regarding hepatitis B, hepatitis C, HIV, syphilis, and malaria. And lastly, I will cover very briefly the selective screening testing for other infectious disease and what criteria should be considered while incorporating it in your testing repertoire. So transfusion transmissible infections, they are broadly categorized into two groups. First is those that result in chronic infections in the donor. And these infections, the donor may be asymptomatic or may have symptoms, but they later feel that they are well, they are okay, but in fact, they have been transmitted to the chronic phase of the disease. But they think that they are okay, so they present to donate the blood, not knowing their infectious disease status. So this is the main category of TTIs, and this include hepatitis B, C, and HIV. The second category is those that result in short period of viremia and which then resolves. The donor may remain asymptomatic or may have symptoms, but the good thing is that there is no chronic phase of the infection. The diseases included in this category is West Nile virus, Zika virus, hepatitis A virus. So what measures should be taken to provide safe blood? There are three measures. First is donor health screening. This is by questionnaire, your universal, by donor history questionnaire, and by physical examinations. A detailed donor history questionnaire is very, very important to identify a healthy donor, because through that detailed questionnaire, the donor could also identify if he or she is at high risk of transmitting the infected blood, so he or she can refrain from giving the donation. The second is infectious disease testing, which I will be talking about. And last is minimizing the window period. So what is window period? Basically, window period refers to the period of immunosilence, which occurs shortly after the individual is exposed to the virus, but the laboratory parameters or laboratory indicators for the infection are not yet at a detectable limit, be it antigen or antibody. So in other words, the labs give negative result, but donor is still capable of transmitting the disease. This is basically the main pitfall of getting transfusion transmitted infection. So this lag periods can be shortened by different techniques used, like for example, if we incorporate viral genome amplification, that reduces the window period, but still there is no test available which would minimize it to 0 level. So in NAT, nucleic acid testing, single unit NAT testing, it can be done in which individual donor is tested separately and just much more sensitive than the mini pool testing, in which we have small number of donor samples. This is, and they are pooled together and they are tested for NAT. So in this slide, I would like to share like NAT testing and nucleic acid testing has the shortest window period, followed by antigen testing, which is much longer than the NAT, and lastly, antibody testing, which has the longest window period. In other words, if we test only the antibodies, then the window period will be very wide, and then there is high chances of transmitting the infection in the window period. So this slide is basically sharing the window periods of hepatitis B, C, and HIV. And you can see in respect to different techniques which are being tested for infectious disease testing. Like in individual donation NAT testing, the window period is shortest. This is followed by mini pool NAT, which I shared, like we pool small quantities of donor samples and then test them as a one sample basically. So it is much little longer than the individual donation NAT. Then this is followed by antigen testing, and then as we go on, the antibody testing has the longest window period. So the chances of transmitting the disease in the window period is highest when only antibody is being tested. This figure shows two window periods like A and B. The A is shorter compared to the B. In A, they are showing the acquisition of the infection and the appearance of the antigen in the blood, which can be detectable. And B is the acquisition of infection and antibody detection in the donor samples. So this is very wide compared to the antigen, and NAT is much narrower than the antigen. From now on, in the slides, I will be talking about the FDA recommendations regarding the transfusion transmissible infectious disease testing. FDA requires that FDA approved screening kits must be used, and those do in accordance with the manufacturer's instructions. And second is they instruct the blood establishments to perform more than one test to identify the infectious disease status to adequately decrease the risk of transmission of transfusion transmitted infections. So in order to accumulate like more than one test, a testing algorithm has been made for each disease or each TTI testing in a sequence of events incorporating both the screening and confirmatory test. So the laboratory should have the protocols in place for both initial and confirmatory testing, and the results should be evaluated in light of both the screening and confirmatory testing to make decision where the unit can be released for the transfusion and what to counsel the donor. The screening assays, they are usually performed, they are the first line tests which are done but false positive results do occur because of the cross reactivities, and the screening assays usually rely on the cutoff values, and any result close to the cutoff value remains indeterminate. So these samples should be tested by confirmatory assay, because confirmatory assay is done on a different testing methodology, and that would like confirm your screening assays results. And also the confirmatory assays used for donor notification and counseling, and then it helps in decision what to be done if the donor has previously donated the blood. So it prospective and retrospective, it helps in both ways. So the question arises, what should be considered while choosing an assay for transfusion transmitted infection? The first point is while choosing any assay for screening of the blood donation, we should see if that kit is intended for screening of the blood donation, because there are kits which are intended for the screening of blood donation, there are others for patient testing. So this should be sought out first, and we should see if those kits are registered by some signatory authorities like FDA, CE, marked, etc. And each donation, FDA also recommends that each donation is found to be reactive by screening test must be further tested by licensed, approved, or cleared supplemental test or confirmatory test if available. But if no supplemental test is available, the blood establishments must perform more than one licensed appropriate test to provide for the confirmation of the result which they are releasing. And then, and also the unit which is being released. Now, something about hepatitis B virus. It's the clinical course of hepatitis B infections determined by the balance between the virus replication and the host immune response. Mostly the hepatitis B virus, it is self-limiting and it resolves, but sometimes it enters into the phase of chronicity. And these are the group of the individuals which are the main concern for the transfusion transmitted infections. The screening assays for hepatitis B virus are hepatitis B surface antigen and hepatitis B core. And FDA recommends that establishments using hepatitis B surface antigen assays should use assays that have the lower detection limit of 0.5 nanogram per ml or less in order to reduce the risk of transmission of disease. So if the assay is not capable of detecting 0.5 nanogram per ml, those assays should not be used for testing of the whole blood and components, because FDA believes that they are not adequate or appropriate for this purpose. Next comes the confirmatory assays for hepatitis B virus. So FDA recommends by the mentioned clause that if hepatitis B surface antigen is reactive, then this should be followed by a supplemental assay to confirm whether the positivity is true positive or it's false positive. So what are the confirmatory assays? One is hepatitis B surface antigen neutralization assay, and other is hepatitis B virus NAT assay with limited supplemental test indication. So regarding the hepatitis B virus NAT, FDA recommends to use the licensed hepatitis B virus NAT, which can detect the infection at earliest stage of the disease. And the cutoff is like less than 100 international unit per ml. Even if surface antigen is non-reactive, FDA still recommend to confirm the non-reactivity by performing the NAT testing with the lower detection limit of less than 100 IU per ml. So there are options for NAT testing, like NAT testing can be done as individual donor NAT testing, or it can be done in mini pool testing that is being approved and by the FDA as well. And there is also multiplex NAT in which the other agents, like in addition to hepatitis B, HIV, and HCV, can be tested by a single multiplex probe. Just a brief about neutralization assay. It's basically anti-HBs. So if any sample containing hepatitis B surface antigen and this neutralization assay is being done on that, the anti-HBs will neutralize a surface antigen if present in the specimen. So it will be neutralized, and this will be considered as positive for hepatitis B surface antigen, and no further testing is required, meaning NAT may be or may not be done. It's not the requirement to do NAT. So either we can use in this flowchart, either we can use hepatitis B virus NAT or neutralization assay for the confirmation of hepatitis B surface antigen reactivity. And if if NAT is positive, then if NAT is positive, then there is no need for further testing. The unit should be discarded and the donor should be reported as positive. But if the NAT is non-reactive and surface antigen is reactive, then neutralization assay is proposed to be done. And if neutralization assay comes comes reactive, then the unit should be discarded and the donor should be reported as positive. But if neutralization assay is non-reactive, then we can report the donor as negative and move the unit to inventory. And same is the case with neutralization assay. If neutralization assay is done as a second line after the surface antigen, then its reactive results, it indicates to discard the unit and positive donor is positive for hepatitis B. But in non-reactive case, we again have to do hepatitis B virus NAT, and then the algorithm is same as for the neutralization assay. A little bit about hepatitis C. So the current public health initiatives aim to identify actively the infected individual to prevent and control the hepatitis C infection and its spread. So FDA recommends that each donation, including the autologous donations, reactive by donor screening test, must be further tested. Donor screening is anti-HCV, must be further tested using a licensed, approved, or cleared supplemental test when available. But if no such supplemental test is available, then blood establishment must perform an additional licensed, approved, or cleared test to appropriately identify if the donor is positive, donor's infectious status. If the result is then negative, the donor should be counseled that HCV infection is not confirmed and is unlikely. But if the result is positive, then no further testing is required and the donor should be counseled as positive. This is a proposed algorithm in which we can see anti-HCV. If repeat reactive, we can do discriminatory HCV NAT. If it is reactive, then no further testing. The SAM is showing no further testing is required and the unit should be discarded and donor should be counseled as reactive for HCV. But if discriminatory NAT is non-reactive, then we should consider for having another serological test by different vendor and do further testing. In such scenario, if the second serological test is also reactive, then it should be considered as the donor should be considered as reactive for HCV and the unit should be discarded. The sources of error for anti-HCV with false reactivity, it's not error. The reason for anti-HCV being positive and the NAT being negative may be that the individual may be infected and it's a false positive result because of the cross-reactivity, or there is a chronic persistent infection and at that time there is low level of viremia which was not detected by the NAT. And then there is a possibility that the infection has been resolved. Now, something about human immunodeficiency HIV. It is recommended that the blood donors who are reactive by screening test for HIV should be considered by supplementary testing before disclosure of HIV status to the donor. So in 2004, the FDA recommended that any donation in which the screening test, which is the antibody to HIV antigen, is positive, the NAT is positive, then the NAT is not essential requirement. But if it is done, it is better for regarding the donor's infection status, meaning what do we need to disclose to the donor? It's better to do the NAT testing. But if anti-HIV is negative on the screening, then FDA recommends that HIV NAT should be performed. In other words, antibody to HIV is negative, then NAT has to be performed. But if anti-HIV is positive, then NAT is not required for the retaining the unit. The unit should be discarded, but it is preferred to do NAT in order to counsel the donor. This is the list of the screening test. As you can see, nucleic acid, antibody, and antigen, all tests are in the list of screening tests. And this is the supplemental test. And all these supplemental tests for HIV have been approved by FDA. So we can use any of them after the screening test. We can, this is a proposed algorithm and it can be designed as per the institutional requirement. Like if the combo is serology of HIV is positive, then we can do a supplemental assay, which is Genius. And if it is negative, then the donor can be counseled as negative and the unit can be incorporated into the inventory. But if the supplemental assay is positive, then the donor should be deferred and should be reported as positive. But if the Genius confirmatory assay is indeterminate, then NAT has to be done. Something about syphilis, as we all know, it is caused by spirochete, Treponema pallidum, and it is usually acquired either after sexual contact with the infected individual. It can have a vertical transmission from mother to child and through the blood and blood component transfusion. So it is in the list of transfusion transmitted diseases. By FDA, it is required to perform serological screening tests for syphilis for each donation. And there are two types of serological assays for syphilis. One is non-treponemal assay and the other is treponemal assay. The difference between the two assays is the non-treponemal assay detects a regained antibody which is directed against the cardiolepin. And cardiolepin is present in a variety of tissues. So they can be false positive. The list includes RPI and VDR in the non-treponemal assay. And the treponemal assay are the tests for the antibodies against the antigen that are specific to the treponemes. And in this, fluorescent treponemal antibody adsorbed assays or TPHA is included in this list. Non-treponemal assays, usually they identify recent infection. They are good for monitoring of the progression of the syphilis, but they are usually initially present, but not for a long term. And then the treponemal assays, like non-treponemal assay, it's better to test the non-treponemal assay with the treponemal assays to distinguish between the true positive and false positive. But please note that both non-treponemal and treponemal assays, they both are antibodies. So early infections can be still missed. A proposed algorithm here for syphilis is if RPR, which is a non-treponemal assay, is positive, then we can do TPHA with a treponemal assay. If it is positive, then the unit is discarded and the donor should be deferred. And while if the TPHA is negative, we can move the unit to the inventory and the donor will be reported as negative. A little bit about the malaria. The transfusion risk of malaria stems from the asymptomatic blood donors, which can be acquired, which can get into the chronic phase of the infection, and hence the donor it's not capable of identifying because donor is basically asymptomatic during the chronic phase of infection. These malarial parasites are stable in the plasma and whole blood for at least 18 days when stored at 4 degrees centigrade, and they can be extended in the frozen state as well. So, but sadly, as this is not the disease of the West, there is no FDA licensed test for donor screening. So, but WHO recommends that individuals with recent infections should be deferred for 6 months after the completion of treatment or full recovery, whichever is longer. So 6 months either from the recovery or from the treatment, whichever is longer. And they recommend to screen all donations for parasitemia using thick and thin blood smears and for the evidence of malarial antigen using highly sensitive enzyme immunoassay or rapid diagnostic tests. These are the WHO recommendations. Lastly, I will just briefly talk about the selective screening regarding other infectious disease testing like HTLV type 1, type 2, Babesia, Trypanosoma, West Nile, and other viruses. So what should be considered if we want to add these disease testing in our repertoire? The question which needs to be answered or which needs to be considered is the infectious agent radially transmissible through the transfusion? If it we have to see if it is endemic in our country, or could this result in severe morbidity or mortality, or is it can be identified through the donor selection which is being employed, and is the kit available, and benefits of screening of an additional TTI in relation to the resource and logistics requirement should be considered, and what might be the impact on the blood supply if such tests is introduced, and are the confirmatory assays are available to distinguish between the true positives and false positives. But the foremost, I think, among all these questions is transmissibility through the transfusion and endemicity, whether it is endemic in your country. That is, I guess, the most important question to be answered before considering to add it in our testing repertoire. So now I would end my presentation, and I would like to conclude that transfusion transmitted disease will continue to evolve forever. And as a blood banker, we should
Thank you very much, Dr. Fatima. That was a comprehensive discussion regarding the TTIs. If there are any questions by the participants regarding this talk, they are most welcome. Nothing. Okay, so we will move on. Our next speaker is Dr. Shabneez Hussain, consultant hematologist at Fatimid Foundation, a well-known hematologist with particular focus in hemoglobinopathies, transfusion medicine, and inherited bleeding disorders. She has also received a grant for point-of-care joint ultrasound in hemophilia from University of San Diego, California. So, Dr. Shabneez, over to you.
Transfusion medicine at Regional Blood Center, Karachi. Today, I'll be talking about the face of hemophilia in Pakistan, and I'll be giving you a reality check regarding this disease in our country on the platform of ICON 2022. So, I have no disclosures or conflict of interest regarding the content I'll be showing in my presentation. To begin with, I'll be talking about the overview of hemophilia. In this, I'll be basically covering how these patients are treated, managed, what are the latest therapies, and how we can actually go forward from there, what is present in our country, what is not there for us. So to begin with, hemophilia, there are two kinds, which are the commonest, hemophilia A and hemophilia B. So if there's a deficiency of factor 8, it's called hemophilia A. If there's a deficiency of factor 9, then the patient is diagnosed as hemophilia B. So in general, hemophilia A is more common than hemophilia B. Around 80 to 85% patients are diagnosed with hemophilia A. So how do you diagnose them? These patients usually present with joint pain, and they usually present in childhood. Either they have a head injury or they may have a gum bleed, whatever bleeding they have. They are diagnosed as they are initially diagnosed as having some kind of inherited bleeding disorder, and then you have to look into it further, that exactly what is the problem with the kid. Then when they grow old, that's when they have arthropathies, because of initial bleed, secondary bleed, tertiary bleed, the repeated bleeding actually destroys the joint. So this is how you actually diagnose these patients on examination. And if it's a really bad case, then you will see these patients with contractures, with flexion contractures, and with terribly immobilized joints in which you have to advise them surgery. So they actually present with all kind of joint problems. And if they have an emergency or a trauma, and this is where you need to treat them on emergency basis. So what's the bleeding severity? So if there are two kinds of clinical cases that usually these patients present with, they either come with a very severe bleed, a life-threatening bleed, and when you start testing them, they either come as severe or moderate. So for severely affected patients, for patients who are diagnosed as severe hemophilia,
their count is usually less than 1%, factor 8 level or factor 9 level is usually less than 1%. But if it's moderate, then it's between 1 to 5%. And if it's mild, it's 5 to 40%. So rule of thumb, patients who have severe bleeding, they can either have a moderate level or a severe level. But patients who only bleed when they have trauma, that's when they are diagnosed. That's when they actually have, when you test them, they come in the milder range. So what I'm trying to tell you here is that severe and moderate clotting factor level can overlap. The clinical presentation can overlap. So when you plan treatment for these patients, you have to really look at the clinical presentation rather than the factor level that you're treating. Sites of bleeding, as I said, they bleed in their joints, they have muscle bleeds, they have GI bleeds, they have head injuries, CNS bleed, spontaneous CNS bleeds. So when they come to your clinic, you need to differentiate between whether it's a serious bleed or it's a life-threatening bleed. So what are serious bleeds? Serious bleeds in which patient's quality of life is severely affected. For example, they have bled into a joint and they can't walk and they have severe pain. If you don't stop the bleeding, these joints can get a major chronic arthropathies. If it's a muscle bleed, of course, they can lose their muscle functions. If they have severe bleed from their mouth, nose, gums, or GI tract or genitourinary tract. So genitourinary tract is something which is serious. So this is how you differentiate which one needs a life-threatening treatment and which one of them can actually survive even if you give them blood transfusions. So in life-threatening cases are something in which you never wait for a factor level. If you know the patient is a diagnosed case, you just give them treatment right away. So what exactly do these patients need? So if I'm a clinician and I'm working in a resource limited setup, for me, if I start thinking of these patients, what do they actually need? They need, first of all, diagnostic facilities. Your patient might be bleeding. They don't know what they're suffering from. So you need to give them diagnostic facilities at least. They need to be diagnosed properly. So I think a major chunk of patients are actually having problem in diagnostic difficulties. Second is they need prophylaxis, because if they do not have prophylaxis is when you give them routine doses of factor concentrates, and you actually prevent them from bleeding spontaneously. So this is actually prophylaxis. Then they need prompt treatment. So if a patient is coming to you on day one of any bleed, you as a clinician need to ensure that these patients are given concentrates on day one of their bleed, and then you can actually transition over to plasma or any other therapy that's present in your center. So prompt treatment means timely treatment with factor concentrates. The other thing is family screening. So family screening, not a very deep area that has been looked into in our country, but to detect carriers, because this disease is being transmitted from the asymptomatic female to their unborn male child. So if you need to stop the progression of this disease in families, so you need to detect carriers in the family. The most preventable thing is oral care. So when you diagnose these patients, what do they need? They need oral counseling. So oral care is something these patients are afraid of. And then when they grow up in their lives, later on, they develop severe bleeds. And for these bleeds, when actually they should be getting treatment for their joint pain and joint bleeds, they are receiving care for their oral bleeds, something which is very preventable. So this needs to be really looked upon. What else do they need? They need to be screened on at least a yearly basis for transfusion transmitted infections, which is hepatitis B, C, HIV. We should really do this screening, because these patients are being exposed to plasma transfusions. So this is what they need. The last thing, which is currently unreachable for them, it's gene therapy. So I'll be explaining each one of not diagnostic facilities, but other things in some detail. So again, the team. So who are the caregivers? The caregivers, the multidisciplinary care, the comprehensive care that we really talk about and we read about. It's a teamwork. It's never a hematologist's responsibility. It's never a physiotherapist's responsibility. We all work together as team. So the hematologist will provide management, The nurse will actually counsel the patient regarding price therapy, how to ice the joint, what to do immediately. The orthopedic will actually look at the affected joints and again will counsel the patient of how not to ensure that these injuries are there. And then the physiotherapist will actually give counseling on how to do physiotherapy. Then the laboratory specialist will actually screen the patient for transfusion transmitted infections, for inhibitors, for any kind of laboratory test that these patients need. A psychosocial expert. So we really need to know that these patients are in constant pain because of the joint disability that they have. So these, when they are in pain, they are unable to work. So psychosocial experts, something very neglected in our setup. We don't do psychosocial counseling. So these patients are actually losing their jobs over their disability, and I think this is one of the most important aspects. You really need to counsel them. Then you have a radiologist. So a radiologist will do their ultrasounds, will do their x-rays if needed, and of course will further prevent the progression of disease. So as I said, it's a teamwork. What do we have usually in our centers is a hematologist, maybe if you're really lucky. And we have a nursing coordinator, we have a physiotherapist, we have a lab specialist. Do we have an orthopedic? No, because no one will devote their time, their special time to these patients, because these are very sick patients, these are non-reporting patients, they can't afford orthopedic care. So you don't have an orthopedic facility, you don't have a psychosocial expert, and you don't have a radiologist. So this is, the gaps are there, and this is where the patient needs the most help. So what to do when you're training the general physicians? You say, okay, so this is the World Federation of Hemophilia's guideline to treat patients. So you have the guidelines, and you also, these guidelines that I'm showing is for the developing country. So there's a guideline for the developing country, but how to implement it? Our problem is not the availability of guideline. Our problem is implementation. Now, once you have the guideline, you know that for treatment you need factor concentrates. You also know that these patients need primary, secondary, and tertiary prophylaxis. If you ask me, I will say our 80% of the patients are somewhere they need between secondary to tertiary prophylaxis. None of them are at the stage of primary prophylaxis because, as I said, they are diagnosed late, and then we are beyond any primary prophylaxis. So either we treat after the onset of second joint bleed, which is secondary prophylaxis, or we treat after the onset of joint disease, which is tertiary prophylaxis. In my career, I have mainly seen patients who need tertiary prophylaxis. So how many protocols we have for prophylaxis? A lot of protocols. We have a protocol by Canada, we have a protocol by Switzerland, we have a protocol by France, and finally something which is really implementable in our setup is the Egypt protocol. But can we follow even that? We can't, because we do not have emergency care for these patients. We don't even have emergency concentrates for these patients. So how are we going to talk about prophylaxis? So as I said, my presentation is on a reality check, so I'll be really talking about reality. So when I say, okay, now I have prophylaxis, how will I actually individualize therapy? So there's a thing called pharmacokinetics. So when I talk about pharmacokinetics, this thing applies in giving these patients factor concentrates. When you give these patients factor concentrates, each patient will have their own way of behaving or utilizing those concentrates in their bloodstream. So some patients will, in some patients, this factor concentrate will get utilized early. In others, it will get a bit late. So in patients who require less of factor concentrates, and if I'm sitting in a resource limited setup, I'll say, okay, so I'm saving my factor concentrates. So is it important for me in a developing country? Yes, it's very important, because PKA, pharmacokinetics, is something which has no cost to it, but all it needs is awareness. So if the doctor is aware that there's a website, you can put in your patient's factor levels after giving them concentrates, and you can actually see what is the time of next dose of factor concentrate for that particular patient. You can easily do it through this. So this is the concept of pharmacokinetics, that when did initially the factor 8 or 9 level was 100%, then it became 50%, then it became 25%, and this differs for each patient, and this is the whole concept. that when do I need to give the factor concentrate again. So this is the WAPS Hemo website in which you can actually put in your patient's readings, and you can actually find out when to give the care again. But how many of us actually know about it? So not many, because this is something we are again at the very baseline level. So now physiotherapy, it should begin as soon as the bleeding has stopped. So we need to have trained physiotherapists for this. Do we have trained physiotherapists? Very limited. I think now international organizations are being focused on getting trained physiotherapists in each developing country, but we do not have it. We don't have many when you consider the number of hemophiliacs in our country, which I will talk about. Now the pain management will what is the reality? Like I said, we need to focus on the reality. The reality is that that majority of the patients, that even I have seen in my experience, and majority of the patients, doctors have noted this, majority are on painkillers, and they're addicted to those painkillers. A lot of patients have been counseled incessantly regarding the discontinuation of the painkillers, so much so that we have even stopped the use of tramadol. Tramadol, Evil, these are the medications, which is being continuously misused by these patients. And can we hold them responsible? We can't, because of lack of awareness, because of lack of treatment. What are these patients to do? How are they going to manage their pain, knowing that they do not even have a job to do? So I think a lot of factors actually make them use these painkillers. So now the international community, or the latest researchers, they have come up with these drugs. So celecoxib is something that these patients can really benefit from, and it can actually decrease the vascularization in their joints, and it can also make them less of an addict that they usually become after chronic pain. So inhibitor is another problem. So for me, if my patient has inhibitor, it's a death warrant for me. Why? Where am I going to get all the bypassing agents, which are just so expensive and so difficult to procure? So inhibitors are, who is at risk? Children with severe hemophilia A. We have seen cases in which the patient was initially diagnosed with moderate hemophilia A, and they turned out to have inhibitors, and we actually looked it up. And I think nowadays the studies are coming up, and they are saying that even moderate patients can have inhibitors, provided that they have those gene mutations. So I think inhibitors it's something that needs to be avoided, and which can't be avoided, because genetic mutation is not in our hands, but this is something the patient should be screened for at a regular basis. So high titer inhibitors and low titer inhibitors. If my patient has high titer inhibitors, I will need loads of factor VIII concentrate to dilute those inhibitors. I'll need loads of bypassing agent to actually bypass the inhibitors. But if it's a low titer inhibitor, I'll again need loads of factor VIII concentrates to dilute those inhibitors. So these are two different things, but it again needs equal amount of money to take care of that patient's pain. What does our developing world do? They follow immune tolerance induction. So if I'm sitting in a resource limited setup where I don't have enough facility to even diagnose my patient, I will not even think about immune tolerance inductions. Immune tolerance induction is something in which you give the patient factor concentrates and you keep on exposing them, and then you hope that someday their body will stop recognizing these factor concentrates as foreign. So yes, a majority will see an improvement, but it is so expensive and it's a prolonged therapy. A few doctors that I know of, they tried immune tolerance induction, but because of lack of factor concentrates, it had to stop in the middle. So something which we can focus on is counseling, and immediate care. Chronic arthropathy, again, it's a story of their lives. They all have affected joints because they have joint bleed, they're not counseled well. I think one of you in my career, I've seen that some hemophilia patients were really into physical therapy and exercise and bodybuilding. Those were the ones who had strong bones and strong muscles. So what happens to them? these patients, when they have pain, when they have acute bleed, they actually lose their hope that something good will come out of it. So this is the point when you really need to motivate them and say, no, you can actually bypass this and you can do more than what you are doing now. So you need to push them towards physical activity, because the stronger the joints, stronger the muscle, the less this complication will turn up in their lives. So now another problem which actually comes in our setup is bleed versus no bleed. So again, the international community came up with this point of care ultrasound. So now their doctors know how to see in the patient's affected joint, whether there's a ligament tear, whether there's a bleed, whether there's synovitis, whether there's an intra-articular growth, anything. They can actually test it with point of care ultrasound. In oral bleeds, so for oral bleeds, again, they have come up with a solution. In in other setups, developed world I'm talking about, they have these laser diode which is there in their outpatient clinic, and they actually they put in a laser beam and they actually freeze the vessels. So this is something which is not available in our setup, but if we are able to really motivate some oral this ENT specialist, we can actually have it in our setup as well. Gene therapy is something which is a dream, but which has become a reality internationally, but it has not become a reality in our setup, because what they do in gene therapy is that they insert missing genes in the patient's genome, and they do it with the help of a virus. It's an adenovirus. Now our patients really need to be hepatitis B, C, HIV negative, and they have to be negative for the adenovirus antibodies as well. So for that, yes, some samples of our patients were taken abroad, but they were all negative. They were all positive for these. I'm so sorry. They were all positive for those antibodies, and gene therapy could not happen. So even if our patients were offered a trial, they were so exposed to these viruses that the gene therapy could not go through. So now coming to Pakistan, what is the current status in Pakistan and where do we stand? I think I've given you glimpses of where do we stand, but now I'll really go in detail. So what is the burden of hemophilia in Pakistan? The WFH has reported that 22,233 patients were with hemophilia who were identified in 2019. Now, of course, this number has gone up, but still the true incidence of disease remains unknown and the actual burden could be higher. And why is this? Because of higher level of consanguinity in our setup, because of scarcity of funds, because we cannot diagnose these patients. So if we don't diagnose them, how can we say that they have hemophilia or not? And also because of poor awareness of this disease. So this is a study. This was actually published from Fatimid Foundation. Again, it will, since this organization is representative from all over the country, this we have an idea. We can get an idea of how many patients are there in our country who are diagnosed and who are not diagnosed. So if you can see, of the total 1,500 patients, only 55% are diagnosed and 36% patients remained undiagnosed. So again, and in a lot of centers, we do not have those facilities that were present in the city, which is fully equipped to do all these tests. So this is the true glimpse. Now, if I go on a ground level, what happens to our patient? How is their day? So this is something which also I had personally investigated, that what happens to these patients when they leave their homes. So when they leave their homes, they actually go to a hemophilia treatment center to get cryoprecipitate. So when they get cryoprecipitate, they go, okay, I got only 4 units of cryoprecipitate. Why don't I go to another hemophilia treatment center and I get some of the rest of the dose that I'm getting? And in spite of doing all the exercise, they're actually getting half of the dose that they should have gotten in the beginning of their treatment. So this is again the reality. So when I had stepped into my hemophilia treatment center, I was having, every other patient was like, I have a joint pain, give me something, transfuse me, infuse me, do whatever, but treat my pain. So when we actually started looking into it, our question was whether there's a bleed or not. And this is where the ultrasound came in. and our doctors and the physical therapists, they started getting trained in this technology because we were saving resources once we identified whether there's a bleed or not. So what treatment options do we have as a nation? We have plasma derived concentrates. We have recombinant concentrates being donated from World Federation of Hemophilia to our patient societies. But those concentrates, all in all, it's very limited. It cannot cater to all the patients that have been diagnosed in our country. So now the current efforts are towards making a registry for the whole country. Secondly, we have tranexamic acid for mucosal bleeds. We have bypassing agents. But where are these coming from? These are coming from parallel trades. So if you tell me a very nice company who gives very state-of-the-art factor concentrates, we will not be able to afford it for our patients because for them, their basic need is something else. So what we do is we get concentrates from parallel trading, and then we actually test it in our setup to see whether it's working or not. And once we are 100% sure that's working, okay, the levels are fine, and the quality control of our equipment is fine. So then we say, okay, now this patient can go for surgery. We can have it in our setup, and you can definitely use this concentrate. This is our daily life. This is how we actually treat these patients. Then the regularity of making cryoprecipitate. So cryoprecipitate, it's like gold for a hemophilia A patient. If you give them cryoprecipitate, they will, they will treat you like you are their savior, because cryoprecipitate is something which is their treatment, and which, if given properly, you can actually replace factor concentrates if you have enough cryoprecipitate in your system, in your setup. So cryoprecipitate, it can actually cover hemophilia A, hyperpigmenogenemia, other bleeding disorders. It's very special. It's a very special blood component. Secondly, there's cryosupernatant. Cryosupernatant is for hemophilia B. For hemophilia B, you don't need a lot of dosing. Every 24 hours, they can have their dosing and then they can go home. But for hemophilia A, you need one dose of cryoprecipitate or factor 8 concentrate every 12 hours, provided you are getting plasma-derived factor concentrates. Fresh frozen plasma, if there's nothing else in the system, the doctor is forced to give fresh frozen plasma to hemophilia patients. So why is this on the bottom of my list? Because this is something I don't want to give my patients because of the large volumes that are going in my patient for a very small dose of factor 8. So for me, as a clinician, I will go for cryoprecipitate, and I'll get it for, I'll give a drive to my technologist to make more and more cryoprecipitate in the blood center. So role of national and international hemophilia society. So there's this thing, it's an international entity, and they are the ones who are actually giving recombinant factor concentrates to the national member organization of each country. Whether it's totally full of developing countries, and they donate recombinant factors from the factories in developing world, and then they donate it to the patient societies in the developing country. So this is called the World Federation of Hemophilia, and it has an NMO, National Member Organization. And for Pakistan, it is called Hemophilia Foundation Pakistan. It is totally made of patients. They are the one who are actually running it, and they have 4 boards. They have one board coming from Karachi, another from Peshawar, Lahore, and Rawalpindi. They have a medical advisory board, and each chapter has its own medical advisory board as well. So what do these chapters do? They actually connect with the hemophilia treatment centers in their region, and then they actually have a liaison with them for sharing of factor concentrates. So till date, it has distributed more than 30 million international units of factor 8 since 2015. So that's a lot of factor. And despite this, I'm saying we've still not fulfilled our requirements. So hemophilia treatment centers is just a glimpse of the hemophilia treatment centers in Pakistan. And like I said, each one is in liaison with the NMO representatives in their region. So as you can see, we have a lot of it in Punjab. We have big organizations in Sindh, and we have some in Peshawar and one in Islamabad. So if you can look at the gaps, so the gaps come in Peshawar, the gaps come in Balochistan. So right now, these are all developing. And now regarding Sindh, the Hemophilia Society in Sindh is now, they have a project in which they have to open 6 hemophilia treatment centers in Sindh. So their next project is Mirpur Khas. They've already established one in Badin, and they've already established one in Nawabshah. So now they are moving forward. So now World Federation of Hemophilia is saying that these hemophilia chapters should make more and more hemophilia treatment centers for outreach purpose. Uh, so what is the ongoing project? The ongoing project is Hemlimbra. So Hemlimbra is something you will keep on hearing about lots of it because it's a latest upcoming thing in hemophilia. It's a prophylactic drug. It's given every 4 to 5 weeks, and it's actually a bispecific antibody with the name of emicizumab, and it binds factor 11A and factor 10A. So what it does, it's totally bypasses factor 8 and stays in the body for 4 to 5 weeks. So very minimal dosing, and the annual bleed rates of patients, they are supposed to decrease. So now they have started, World Federation of Hemophilia has now started distributing it to developing countries. And from our country, 100 patients are on this trial, and from SIM, 35 patients are on this trial. So this has to be seen whether this can be donated to our patients on a long-term basis, and maybe we can, maybe more patients can be enrolled in using this bispecific antibody. So to summarize, what is our reality? Our reality is diagnosis. So why do I keep saying diagnosis? Because in outreach areas, the biggest problem is diagnosis. We don't know what that patient is suffering from. Secondly, even if we know what these patients are suffering from, they have limited factor concentrates, and they don't have timely access, and the cost is high associated with these concentrates. But there's always a way out. There's always a shining light, and when they say that there is always a ray of hope, the ray of hope is regional blood centers. So these regional blood centers, they have the capacity to make cryoprecipitate, and now at least Sindh government has allowed. I'm not sure about Punjab or Balochistan, but Balochistan, yes, I know they are doing it. We are all giving cryoprecipitate to this part of to this particular group of patients, because we make it, we have the capacity to make it, and we are allowed by the government to help these patients. So I think it's a ray of hope, and a lot has been done, a lot has been shared, and this is something to look forward to. And I think another way forward for these patients would be plasma fractionation in our country. So now things are looking up. Even the International Society of Blood Transfusion is now having CMEs, like resource plasma fractionation in resource limited countries and everything. So now we're actually thinking about having this kind of setup in our country. So that is a ray of hope. The other thing is majority are not on prophylaxis. So what to do? What we can do is that we can actually say that to these patients is that get your cryoprecipitate or cryosupinated infusions on days when you think you have the most physical activity. Can it be done? It can be done. And a lot of patients are automatically doing it. We are not telling them to do it, but they actually made their own lifestyle, and they said, okay, so if we get these infusions once or twice a week, we are actually fine. So this is something their need drove them to it, but actually it is prophylaxis. The other thing is joint pain. Every joint pain and swelling is assumed to be a bleed. So if you do not, what is our drawback? Even though there's a thing called point of care ultrasound, not many people know about it, and there is a need for training people for this specifically, because it can actually reduce the amount of factor 8 and factor 9 in plasma these patients are receiving. So again, trainings need to be organized for dedicated physicians and for dedicated physiotherapists who can actually take these patients forward. So lack of multidisciplinary care, again, for me personally and for other setups also, there is a multidisciplinary care available, but a lot of effort is needed to actually make the other doctors understand how important these patients are, and they also have a right to survive. Secondly, importance of physiotherapy is not understood by majority of the patients. So more and more counseling, more and more awareness. Oral bleed, again, if you counsel them when they are diagnosed, at the time of diagnosis, if there's counseling for oral bleeding, I think we have these patients covered there. Then in rural areas, physicians need to be made aware of. I think SOPs for clinical treatment of these patients should be given to them at the time of training. so that they know how to take care of these patients, and it's so not difficult, it's so very easy. So with this, I would like to end my talk, and thank you so much, and I especially thank Indus for this platform. And thank you so much. Thank you so much for the opportunity. Thank you.
Thank you very much, Dr. Shabneez. Uh, this was very enlightening regarding the hemophilia and ground realities which we are facing. If there are any questions by the participants, they can kindly ask via the chat. Okay, so our last speaker of today's this session, Dr. Bushra Moiz. She is consultant hematologist at Aachen University Hospital, a senior hematologist with a special interest in hemoglobinopathies, transfusion medicine, and coagulation disorders. She has published more than 80 papers in national and international peer-reviewed medical journals. So, Dr. Bushra, you may start.
Assalamu alaikum, everyone, and a very good morning and afternoon or evening, whatever time of the world you are. So I'm very thankful to ICON 2022 Organizing Committee for giving this opportunity to me. I'll be talking on a very important topic, which is hemovigilance, role of Hospital Transfusion Committee. And introducing myself, I'm Dr. Bushra Moiz. I'm professor in section of hematology and transfusion medicine at Aachen University Hospital. I don't have any conflict of interest for this presentation. So let's understand the term hemovigilance first. Hemo is a Greek word which means blood, and vigilance is a Latin word which means watchful. So when we define hemovigilance, it is a set of surveillance procedure of transfusion chain from collection of blood component to follow-up of its recipients, intended to collect and access information on expected or undesirable effects resulting from therapeutic use of labile blood products and to prevent their occurrence and recurrence. The important keywords I've underlined in red so that you can understand that hemovigilance is all about surveillance. And what are we surveillance? We are doing the surveillance of the transfusion chain. We are doing the surveillance of the donor, the process, and the recipient. And the main aim is to provide some help to and oversight to the blood bank and to the hospital so that adverse effects do not happen. This is the main purpose of hemovigilance. So looking at international models, it was started first in 1993. So it is not a new concept. France was the pioneer in starting hemovigilance in their country. And it was then later on taken by the UK in the European countries as SHORT, which is Serious Hazards of Transfusion in 1996. And it made them voluntary in contrast to the France where there was mandatory reporting of the adverse reaction of the blood products. Then it was later taken up by the European Blood Directive in 2002. There are various international global models are available. They are taken up by the European countries, by the Asian countries, Canada. And so the entire world is talking about hemovigilance and have made societies and regulations to control hemovigilance. So the International Hemovigilance Network was formed in 2009, and it is coordinated with International Society of Blood Transfusion, ISBT. And the main purpose is to provide surveillance at the international level. Because if you see, initially it was started at very limited scale in the European countries, and then some countries take it over as their own initiatives. But then International Hemovigilance Network came into existence to invite all the countries for their participation. So Pakistan became member of the International Hemovigilance Network in 2013. And the key purpose was to set the targets, to achieve some targets, and to plan for the future. And I must say that this hemovigilance is very good in maintaining the data for the donor prevalence or seroprevalence of the transfusion-transmitted infections. But otherwise, because it is dependent upon the blood bank providing the data to them, so the data is somewhat sketchy and does not give entire picture for the country. Hemovigilance at AKU Blood Utilization Committee is the Hospital Transfusion Committee, or HTR, of AKUH. We call it BUC. And I will tell you the hemovigilance story at AKU, that how it happens and what are we doing at AKU, and perhaps there may be some indicators, quality indicators, or some lessons which we have learned over a period of time, which may help others to build up their own hospital transfusion committee, which they can work solidly or consolidated their performance in order to achieve certain targets over a period of year. So this blood utilization committee of our Khan University Hospital, it was founded in 2000. We have monthly meeting, and we provide annual report to our leadership, which is Joint Staff Committee, and to the medical director. And this now medical director is called CMO, which is Chief Medical Officer. What does it do? It identifies and resolves blood-related issues and maintains records of quality indicator, which I'll be showing you throughout my presentation, that what are the quality indicators which we measure and what are the initiatives which we take in order to improve the situations. So these are the member representative. The purpose of showing this, the member representative of HTR, is not to show you the names, but to show you the disciplines where they belong to. So the idea of Hospital Transfusion Committee is to have those members on board where there is blood utilization. And these are the key holders, for example, surgery, medicine, oncology, cardiac surgery, OBGYN. These are the solid people who use blood units all the time, and therefore they are included. Their participation and buy-in is very important, because whatever we say, it has to be implemented in these areas. So these are actually the people who are ambassadors of Blood Utilization Committee, and they ensure that whatever Blood Utilization Committee is asking for, that is implemented in their area. And obviously the chair and co-chair are usually the people from the people who have understanding of hematology and blood banking. So it's usually from traditionally, it has always been a hematologist who is chairing that. And then they are representative for the blood bank. Nursing are very important for here, because in our hospital, it is the nurses who transfuse blood products to the patient, and they are the one who monitor the transfusion reactions. So their representation is also very important. And it is also important to have representation from anesthesia, because they are the one who order blood products for patients who are undergoing surgery. So it is desirable that we are all on one page and we understand what are the targets which we have to achieve for over a period of year. And then we seem to have, in the end, we discuss our goals, whether we have achieved those goals or not. So the main aim of hemovigilance is to improve safety of blood transfusion. In a nutshell, this is the aim. But in order to do that, we have to look at three items. We have three buckets to be where we are doing hemovigilance. One is the donor, second is the product or the process, and third is the recipient who is receiving the blood transfusion. So the scope of BUC, or the terms of reference of BUC, it spreads over all three areas. And one thing I want to tell you here, that all these data, they are collected by the blood bank. The quality coordinator of the blood bank collects the data, they provide this data to BUC for discussion, and then we look at whether we are going in the right direction or not. So in a way, this BUC is providing an oversight of the data for the correction and preventive actions, which is provided by the blood bank. So as far as the donor is concerned, we look at the review adequacy of blood supply. If there's any shortage of blood supply, then we look at it. We look at the contingency plan to address the shortages of blood products through doing clinical audits that where we can conserve the blood products. Then in the process, we look at, we review the wastage of the blood unit, which is very important to avoid the wastage of this precious product. BUC also helps in development of transfusion guidelines. They develop audit criteria. Then any new transfusion related product or any new activity that is required by the blood bank, that also goes through BUC. For example, in 2011, blood bank wanted to start nucleic acid testing on the blood product. This was approved by BUC, looking at the pros and cons. So there's a system of oversight or overview that is provided by blood utilization committee to help blood bank to achieve its goal. Then it also looks at the transfusion service workload and adequacy of resources, whether the staffing is okay or not. They review the hospital-wide policies as well. For example, the administration of blood product is a policy that is made by nurses, but it is reviewed by the blood utilization committee as well to see whether it is appropriately made or not, or they have made it according to the standards that are required by the blood bank. Then the last bucket, which is recipient, the blood utilization committee review the transfusion reactions and also monitor the transfusion practice through audits. So what is the oversight that is provided by blood utilization committee for the donor hemovigilance? So this is the data which I'm showing you for 2020. Obviously, the blood that was collected in 2020 because of the COVID pandemic was less compared to that we collected in 2019. And we observed that there was a 20% drop in the collection of blood product because of the COVID. And there were numerous reasons for doing of that, because the donors were afraid. Initially, they were afraid of coming to the blood bank because of the risk of contracting COVID infection. Nowadays, we are faced with the challenge of collection of the blood because now the donors are getting vaccinated, and we have to defer them for 2 to 4 weeks at least before they can donate blood. So there are new challenges which we are facing in collection of the blood unit. But here I want to emphasize that in AKU, the main supply of the blood bank is exchange donation. That is, the donors who come they donate blood for their relatives, for their friends, and they are the one who actually form the main or the major bulk of the blood donors. That is exchange donation. And what we observed that our exchange donation in 2020 compared to the previous year, it remains the same at 97%, but we were badly hit in the voluntary donation. It was previously, this number was almost up to 12% or so, and now it was reduced to 2.5%. And perhaps this was the reason that which I talked previously, that why the voluntary donations were decreased. And we need to look at that, how can we make our voluntary donors comfortable so that they keep on donating blood for us. Blood screening of the reactive products. Blood utilization committee also looked at the total donations and the reactivity. It has been historically 2% almost for a number of years. But the role of blood utilization committee is to see that it does not go beyond 2%. If the rate start increasing, then we have to take strict measures that why there's an increase in the reactivity of the blood product and what measures should be taken to reduce it. And among the transfusion reactions, we see that because we are doing nucleic acid testing as well, so we see that serologically as well as nucleic acid testing, hepatitis B virus is the most common virus which our blood donors are carrying, followed by hepatitis C. The other viruses like HIV, that is not that common, fortunately, and we very the VDR, the syphilis, in serological seroprevalence for the syphilis and for the malaria, that's very infrequent in our situation. This is perhaps because of the verbal screening which we do for the blood donors, that they are screened before they can donate blood, so that because of the deferral of the donors at the verbal screening, that is perhaps the reason that why our blood products are so clean and we get very less reactivity in the blood product compared to the other donation centers which are reporting a very high incidence of reactivity of the blood product from Pakistan. The another problem which we see is, which we published as well, that majority of the reason for deferring the donor was anemia. And I want to share with you here that at AKU, majority of our blood donors are males. It's only 5% or so that are female donors, but majority of them are males. So it's very worrisome that we are seeing anemic males who cannot donate blood. We know that thalassemia is common in Pakistan, and which may be the reason for mild anemia in these donors. But then we have not looked into, but this is an important thing, that we need to follow these donors. We need to we need to have a plan for these donors, because we need to maintain a pool of donors, because we want these donors to come back after their iron deficiency or vitamin B12 or folate deficiency has been compensated. Then we need these donors to come back. But right now we don't have any plan for following these donors who are anemic. But this is worrisome that majority of our blood donors who were deferred had anemia as a reason. And you can see the big block, which is the proportion. You can see majority of them were anemic, and that's why they were deferred. And then there were other reasons, which were the diseases because of which they were deferred. We also monitor the adverse reaction of the blood donors, and this is through our blood bank information system. So any donor who has any adverse reaction, for example, he faints because of vasovagal syncope or has vomiting or feels dizzy, that is reported, that is collected into the system. And we also monitor the adverse reaction for the donors if they're increasing or they are static. So we have noticed that majority of the adverse reaction for the donors which happen are the vasovagal syncope, or they usually say it in the form of whether they are having ghabrahat or they feel like dizzy. So majority of the time we find that this is, and then we need to have measures for doing, for reducing this adverse reaction for the donors as well. Also published this data, which is regarding estimating window period donations for HIV, hepatitis C and B through nucleic acid testing in southern Pakistan. And here I want to show you that our donors are showing a positivity for the HCV and Hep B, which is very high per 100,000 donations. Which means that if we don't do nucleic acid testing for the donors, we may be able to transmit these infections to the recipients if we do only seroprevalence. So what we did was, in order to save the revenue, we took only the seronegative donors and we tested them for the nucleic acid testing, and the data shows that we have a good incidence of having residual infection. For example, for HCV, it's very high, that of 13 or of every 14,000 donors, one will be carrying HCV. And for hepatitis B, every 11,000 donors, one will be carrying hepatitis B, and that will be missed if we do only the serological testing. So what were the recommendations of the BUC after looking at these? We need to reduce the adverse reactions which are happening in the donor, and we also need to work on the anemic donors. Perhaps this will be made as the targets of future work in 2022, that we need to reduce the adverse reactions and we need to work on the anemic donors. Anemic donors do not donate blood, they are deferred. But it is important to take care of them because these our donors are precious. If they have come to donate blood, then that means they may be, if we convince them, they will come again. But because of the anemia, they are temporarily deferred. So if we keep a track on them, if we provide them with the guidance that how to correct anemia, perhaps we can repool these donors, and we can help them in maintaining their integrity also. Because when you defer a donor, this has a very negative impact on the donor. So we need to help these donors so that we are able to maintain the pool of our precious healthy blood donors who can donate blood to us. Having done with the donor hemovigilance, I'll talk about the product of process hemovigilance. So if you look at, this is data which I'm showing you for previous years, that regarding the sampling errors, which means that how many wrong addressograph happen. And you can see that initially they started with a high number, then they reduced for the low numbers. And that is because of the various improvement that were done by BUC. And I'll show you what we did for avoiding the sampling errors which were rampant in the previous years. The Blood Bank Utilization Committee also looked at the blood unit wastage, because obviously we don't want our precious products to be wasted. And they are wasted because when they are unused units which are returned from the floor, they may not be at the required temperature, and therefore they may be wasted. So what we have done is we have made a rule of 30 minutes. That is, within 30 minutes, the unit should be started for transfusion and after dispensing from the blood bank. And if there is no transfusion, that unused unit should return to the blood bank within 30 minutes. This is called 30-minute rule. And we follow this through our computer system. And what the blood bank does is we issue letters to the floor or to the doctors where the transfusion is made out of hours, like where 30-minute rule is not offered. Because if they don't follow that, we cannot restock those blood units and they will be ultimately wasted. So you can see the number is less here, 3 to 4, and this is because of the very strict hemovigilance that is provided by the Blood Utilization Committee, that the blood products should not be wasted. And the floors have to monitor the rule of 30 minutes. And they have to return the blood unit within 30 minutes so that it is not wasted. Then there's another indicator, which is Rbc expiry. And you can see a lot of, in the numerator, you can see the units that were collected, the unit that were wasted in 1 quarter. And this expiry happens because if the units sit in the refrigerator for a long period, beyond 35 days, obviously it will be expired and wasted. So this was realized when we started looking at this indicator. We realized that what we were doing at the inventory level, the rule which we made there was first to go in is the first to go out. That means what was happening in the blood bank was they were releasing fresh units of blood, while the old units were sitting on the shelves for a longer period of time. So with this rule, first to be in is first to be out, help the blood bank inventory to reduce the expiry rate, and you can see that it dropped down from 3 to 2, but still the target goal is less than 1%. So we need to further work upon it, and we need to reduce the expiry rate to less than 1%, because in this, the units which are expired, they are precious units also, which are O negatives. So we need to monitor this expiry rate very well, so that we don't expire our precious units, and the target is less than 1%. This is one ratio, which is cross-match transfusion ratio for the elective surgeries, where we need to improve this ratio, because cross-match transfusion ratio for elective surgeries means that the number of units that were transfused, which is this one, over the number of units that were transfused, we make it a ratio, and the ratio, ideally the ratio should be less than 2.5, which is given by the British Society of Hematology guidelines. But if you see that it has been always above, this is the red line, and it indicates the transfusion in the elective surgeries has been more over a period of time. This was discussed in BUC, and what we have done is, because the elective surgeries include all kinds of surgeries that are happening in neurology and OBGYN and cardiac surgery. So what we did was, instead of looking at the generic ratio, we looked at the ratio surgery wise. And there we found that it's mainly the vascular surgeries and the orthopedic surgeries that are doing over cross matching. So now the blood bank, this data will be discussed in the next blood utilization committee with the members. And what we have decided as the protocol is, the chair of the blood utilization committee will send letters to these surgeons who are over cross-matching the blood products, so that we will be able to know what were the reasons. Perhaps we can help them in setting the targets. And here I want to tell you that the blood utilization committee actually helped in making MSBUS, which is maximum surgical blood ordering schedule, and the number of blood units that need to be defined, that need to be cross-matched, have been defined for each surgery. For example, in C-section, you don't need to arrange blood product. For lab chole, you don't need to arrange blood units. And for the cardiac surgery, you need to cross-match 4 units of red cells. This has been made looking at their own practices, and this has been calculated, and MSBS has already been made. But despite that, these are the numbers which we are looking at. So now we need to be more, I should say, we need to have a non-punitive approach, but we need to convey our message to the surgeons that they need to reduce the cross matches of the blood units, and perhaps there may be some issues in the understanding or apprehension of the blood unit, of getting the blood units at the time of emergency that they're doing that. So we need to have an open dialogue with these in order to reduce the this ratio. Then incident reportings in 2014, you can see that there were certain incidents that were reported by the blood bank, and certain were received by the ward. So each incident we look at very critically, and we close it, and we learn the lessons from them, and then we try to close these incidents, that what has happened in the, why it happened, and what measures can we do in order to prevent it. We also look at the QC monitoring, which is done internally by the blood bank. For example, here I'm showing you the data of the platelets QC. We look at the pH and the other, for example, the hematocrit of the packed cells and factor VIII level of the cryoprecipitate. And if you look at it in this one of the blood product, for example, in June 2014 was found to have bacteriological contamination. So we usually look at the indicators, only at those indicators which have non-conformities, and then they look into that what action needs to be done in order to improve the situation. So when this was looked at in detail, it was found that this was a bacteria and that was there, which was picked up, but it was an environmental bacteria and it was not repeated on the same culture. So microbiology department gave us the input that this may be contamination during culture process, and this may not be a bacteria that has been transfused into the patient. So what we did was we did quarantine the FFP, and we take care of the platelet incubator. We asked the blood bank to provide us with the culture results of the platelet incubator, so that and then it was found that there was no growth in that. So that's how the surveillance is provided by the blood utilization committee and oversight of the functionalities of the blood bank that are done by them. CAP surveys, these are monitored by the blood bank themselves, and usually the blood utilization committee does not have an input into this because already they are doing a good job of 100%. Cancellation of the blood product. This is a sore issue where a lot of blood products have been canceled. This data I'm showing you for the year 2020, which is the pandemic year, and we need to look at whether this is a continuous in the post-COVID era as well or not. So number of the time blood was arranged, and you can see that it was majority of the time it was red cells. 62% of the time it was the red cells that were cancelled by the physicians. So we need to look at that what should be done in order to reduce the cancellation of the blood products. So the lessons that were learned, the blood utilization committee, the functions, the actions which BUC need to take is to reduce the expiry of the blood units, to bring it down to less than 1%, this to reduce the CT ratio from 3 to 2. This is again an objective for 2022, which we need to do, and the cancellation of the products. That is, we need to reduce the cancellation of blood products from 40% to down. Although these products are being reused for other patients, but this means a lot of unnecessary work on the part of the blood bank, which means the blood bank will have the stopping issues because of that. So we need to reduce the cancellation of the blood products, which is done by the physician. We need to very actively and participate in education of the physicians that when to order the blood product for cross-matching, and so that because of the appropriate action is required from the physician side, so that cancellation of the blood products can be reduced. And finally, about the recipients, what are we doing for recipient hemovigilance? So in the last year, we transfused about 50,000 blood products to the patients at AKU, and majority of them were red cells and platelets. Plasma and cryoprecipitate were less transfused to the patients. And if we look at if you look at the range, the blood products arrange over 89,000, and the transfused products of 52,000. Again, that is what I was talking about, that about 40% of the blood product were cancelled and they were not transfused. So this is an extra work on the part of the blood bank and needs to be looked at very critically. Transfusion reactions that were reported in 2020, there were mild reactions that were reported. Majority of them were febrile reactions and allergic reactions. Transfusion-associated dyspnea and transfusion-associated cardiac overload were less common. Hypertension was also noted. And then there were some reactions that we couldn't put into any category, and they were put into not otherwise characterized NOC category. So we look at the quality indicators into mild, moderate, severe. severe reactions are trolley or sepsis or very high allergic reaction, anaphylaxis that happens, or wrong blood transfusion. These are also monitored, and we look at them very closely. That what are the learnings from this, and how can we improve these transfusion reactions? So we published this for complete transparency. We also published this transfusion reactions in one of some time back. And when we looked at it, again, we found that it's mainly the fever and allergic reactions that are happening in our patient. And although the transfusion reaction is within what has been reported worldwide, but we need to look at that how can we reduce the febrile reactions and allergic reactions that are happening to our patients. And these are the evaluation of ABO incompatible transfusion reactions that happen. And it was found that majority of the time, it was failure to identify the correct recipient at the bedside that lead to ABO incompatible transfusion reaction. And sometimes it was the wrong addressograph or the mislabeled sample tube that was sent that led to the wrong transfusion. And I will tell you what measures we took for this, because this is the data up to 2012, and this has been considerably improved in the recent years. For example, we did not see any ABO incompatible transfusion reaction from 2017 onwards till now. And how did we do it? The nurses were reinforced to do a final bedside check. There are 2 nurses who check the compatibility before transfusing the blood product to the patient, so that make sure that it is the correct recipient. And second thing which actually helped us is the bedside ABO typing. This is done by the nurses. So nurses do check the blood group of the patient at the bedside before transfusing. And if the this blood group, which is done by them, does not match with the result which is written on the blood unit, then they inform the blood bank, and the blood bank look into it. So in the previous year, at least twice the nurses identified that the blood unit group is not matching with the blood group which they have identified, and finally it was found to be the wrong blood in the tube. That was the reason for this, and we were successfully able to prevent the transfusion of ABO incompatible blood into the recipient. So this is a very successful story, and this has helped us a lot in preventing in ABO transfusion of the ABO incompatible blood products. The other thing which we monitor is the deviation for the recipient. For example, the head of the blood bank, on the daily basis, they looked at that what are the deviations from the normal, or what are the non-conformities. For example, if an Rh positive product has been released to Rh negative patient, or patient has been released a blood product before completion of the serological or the serotyping for the transfusion transmitted infections in emergency, if it has been released. So these are some of the things that are automatically come into the system, and they are released to the people, the blood bank manager, and to the doctor who is looking after the blood bank, and they check it on the daily basis. And we need to see if we are releasing the blood product before completing the compatibility, for example, the cross matching, or we are releasing the number of units before doing the serological typing of the patient, of the blood unit, then we need to look at that how can we reduce these dispensing of the blood units that are happening without completing the blood bank processes. So the deviation rates are also monitored, and we look at the data that how these can be improved through proper education of the physicians, and through proper maintaining the proper inventory so that we have enough surplus blood that can be released without completing the testing for the infections to the patient. That usually happens in, for example, in this case, the platelets were released without completing the reaction because, and you can see that the patient had upper GI bleed, patient platelet count was 35. So it was approved for deviation because it was an emergency setting. Therefore, the blood bank allowed, the head of the blood bank allowed this deviation to happen. So this data is also monitored for the recipients. Rh-Ig monitoring is also done. For example, those females who are Rh negative and they have given birth to a baby who is Rh positive, for example, here you can see the baby born is Rh positive to a negative mother, then we need to make sure that Rh immunoglobulin has been issued to them and that within the 72 hours, the cutoff time. So we monitor this data as well to see that there are non-conformities or not, so that the recipients, the O negative females can be protected from hemolytic disease of fetus and newborn in future. This is also some data where we do the hemovigilance to protect our recipients who are Rh negative mothers. In summary, I've shown you a number of data for donors and processes and for the recipients. So in the summary, I want to say that hemovigilance ensures safety of the donors as well as recipients. It requires training of health professionals of the varying levels. I must say here that the data which I'm showing you is, it has matured over a period of time. Those blood banks who are interested in doing, forming a hospital transfusion committee and maintaining the data and providing vigilance, they should start from some simple collection of data like transfusion reactions that are happening, so that, that over a period of time, the data is going to get mature and you can add many quality indicators. But the work which I've shown you, that was not done overnight, and nobody can do that. If you notice that this blood utilization committee started in 2000, so it's been 20 years since we are working, 2 decades of work. So, but obviously you can learn that what vigilance can be provided from the blood banks for the hospitals or institutes who are maintaining these data, where the hospital transfusion committees are active, they're working, and they are bringing some good change in their, or a positive change in their practices. So these are the lessons that can be learned from the hospitals who are maintaining a good hospital transfusion committee. But one thing I should say, which is the last line of my conclusion, that approach should be supportive. It should not be punitive, because if you, the data which I've shown you is provided by the nurses, by the doctors, by the people who are working in the blood bank, and by the residents, interns, physicians, consultants, surgeons, all over. But if we become punitive, if we start looking at them and start giving, ridiculing them, mocking them, or punishing them for the wrong errors, then the culture, the safe culture will not happen. They will not share the data. They will start hiding the data. They will not, because if a ABO incompatible transfusion has happened, they will not tell us. This is only when there is a culture of equity and when there's a culture of self-centeredness, then only the people will tell you the problems they are facing, and then the blood utilization committee or hospital transfusion committee can provide a supportive role for the hemovigilance, which is actually the ultimate role, is providing patient safety. So I end over here, and this is these in the summary. I also want to tell you that the indicators which you have shown for that IT support is imperative, and if good IT support is not there, then that means the data needs to be collected manually, which in itself is very tedious, time consuming, and there are chances of errors as well. But if IT support is not possible and the blood bank information system is not monitoring its data through electronically, then you have to have some way of dealing with the collection of that data. So it can be done manually, although there are pros and cons of doing that. But the blood banks need to start from somewhere in order to start the quality indicators, in order to monitor the quality indicators for patient safety. This I have to discuss initially that the patient vigilance, we need to reduce the FNHTR by leukoreduced blood products. And we also need the barcoding for recipient identification in order to improve the final bedside check. Thank you.
Hello. Assalamu alaikum. Thank you very much. I would for from all the speakers. We would like to have our question answer session now. This is Dr. Saba Jamal. We have our speakers online. Some of them, Dr. Shabneez is there, Dr. Fatima, Dr. Neelum. Anybody else? और नहीं है ना, तीन ही लोग हैं ना अभी, बाकी जा चुके हैं. ये इसमें Dr. Uzma is at home with COVID. So I think she cannot attend that, and Dr. Bushra bhi nahi hai. Magar Dr. Shabneez, Fatima, Meraj. को आ रहा है? एक सेकंड अभी ऑन नहीं कर. हेलो, आवाज आ रही है? ठीक. Okay. Sorry for this disturbance. Glitches, online glitches. So I would like to close this very important topic of blood safety, and it is actually really like connecting the dots. Blood transfusion is a very important topic of hematology, and it has, I think, it's more so important in Pakistan because it's very neglected. Recently, in the last few years, because of the safe blood transfusion program and the interest of the German Development Bank, etc., this has been become to the forefront. And but we need to talk about it more and implement it more. And I would like to thank all the attendees, people who have taken out the time. All these talks will be put up on the website. So you can, those who are unable to see, listen to most of them can go back and listen on the, or view it on the website. And I would like to thank the speakers. Dr. Shabneez really gave a very enlightening talk on and giving us the reality check, which is quite poor for hemophilia patients. She's been on the forefront of trying to change the hemophilia care in Pakistan, and hopefully, inshallah, in future, things will improve better in most of the centers. And she was able to discuss and address many points. Dr. Bushra gave a very enlightening talk on hemovigilance, which needs to be implemented in most of the hospitals and is still a very neglected portion of blood transfusion medicine. Dr. Uzma talked about the challenges that were faced during the COVID pandemic for regarding the collection of voluntary blood donors. And then Dr. Neelum gave a very discussed a topic that is really untouched, which is good manufacturing practices. We think about good manufacturing practices for drugs only. It's not. It's the entire process of blood banking. And then Dr. Fatma Meraj gave us this talk about TTI testing, what we already know and what that needs to be addressed, especially the supplemental testing. And these are the things that we have addressed in Indus recently in the last year, and so have implemented the supplemental testing of almost all the tests and all the diseases. And I think this was very enlightening. Thank you very much. And inshallah, see you soon at the Pakistan Society of Hematology meeting. Thank you again, all the participants and all the speakers. Thank you very much.